Raegan's presentation, screening various peanut Arachis hypogaea Linnaeus genotypes for aflatoxin contamination using in vitro seed colonization of Aspergillus flavus.
All right, thank you for the introduction. So yeah, today I'm going to be talking about my research on aflatoxin in peanut. So for those of you who don't know, I'm just going to give a little brief introduction to aflatoxin. So it's a fungal toxin. It's produced by different Aspergillus species, and this is mainly flavus and parasiticus. It can cause diseases in both humans and animals, and it's a really big problem because it can happen pre-harvest before peanuts are harvested in the field, or it can happen post-harvest during storage of peanuts. And the production of aflatoxin can be extremely sensitive to environmental conditions, especially high temperature and drought stress. So those are all some challenges with studying aflatoxin. So just to give you guys an idea of how potent aflatoxin is, I thought I would put it on a scale of some common carcinogens. So alcohols down here at the left, it's not very strong, followed by different types of food additives like aspartame. And then you move on to pesticides and formaldehydes, which are very strong carcinogens. And then all the way over here to the right, you have aflatoxin B1, which is the single most carcinogenic naturally occurring substance that we know of. So it's very strong and very dangerous. So how does aflatoxin get to consumers? So Aspergillus can infect a wide variety of crops, not just peanut, but also it's a really big problem in corn as well. And after the crop's been infected, if environmental conditions are right, then aflatoxin bees can be produced. And then if humans consume those crop products, then they can become exposed to aflatoxin. But also peanut and cornmeal is something that's a key ingredient in animal feed. So if infected peanut or cornmeal makes it into animal feed, then animals can consume the aflatoxin and they convert it to what's called aflatoxin Ns in their bodies. And if we eat the poultry or the cattle, then we can become exposed to aflatoxin in that way. And that can lead to a wide variety of different health issues, including growth stunting, aflatoxicosis, which is like an enlargement of the liver, immune suppression, and I think one of the biggest issues with aflatoxin really is liver cancer. Is strongly linked to aflatoxin exposure. And so it's a worldwide problem, not just here in the United States. It's estimated that 4.5 billion people are chronically exposed to aflatoxin in their diet in some way. And it's the causal agent of up to a third of the world's liver cancer cases. And just in the United States, it causes over $1.5 billion in losses every single year. And so last year I kind of presented some of my aflatoxin work, and it was kind of based off of this original study by Walid Karani. And so what this study looked at was different peanut genotypes. So you can see along the y-axis here are different peanut genotypes, and along the x-axis, sorry, and along the y-axis is the amount of fungal growth that was found on these different peanut genotypes. And what you can see here is there really wasn't a statistical difference in the amount of fungal growth between these genotypes. But then when they were assayed for aflatoxin contamination, we saw a difference in some of the aflatoxin levels on these different genotypes. So along the y-axis here is aflatoxin contamination levels. And so you can see this genotype here, ICG 1471, was showing some aflatoxin contamination resistance, whereas this genotype over here, Florida 07, was showing a lot of susceptibility to aflatoxin. And so whenever we map the ratio of fungal growth along the x-axis here and aflatoxin along the y-axis here, you can see ICG 1471 right here has a lot lower ratio of aflatoxin to fungal growth, whereas Florida 07 here has a lot higher ratio of aflatoxin to fungal growth. And so what we did after this study is we developed a population, a recombinant inbred Lyme population from these two genotypes, ICG 1471 and Florida 07, and we selfed those for several generations, and we started assaying for aflatoxin in the F6 seeds. So this is an actual picture of all of the seeds that I assayed for aflatoxin. So we did 4 replicates of experiments on all of these F6 seeds to look at their differences in aflatoxin contamination levels. And so what we found with that study is out of all of these seeds that we assayed, we found 4 that were continuously expressing the aflatoxin resistance. And so these are ones that we actually did some genetic analysis on, and we decided to do some further evaluation for their aflatoxin contamination resistance. And so that's part of this study that I'm presenting today, is we included these 4 recombinant inbred lines in a larger study where we wanted to evaluate aflatoxin contamination resistance in multiple genotypes. So we wanted to provide evidence for the contamination resistance in these recombinant inbred lines, but also screen some additional genotypes that may have resistance to aflatoxin. That way we can provide evidence for some new sources of aflatoxin contamination resistance that we can use. And so this is a picture of all the genotypes we used in this study. So they were genotypes from that recombinant inbred line study, which were those 4 resistant lines, along with 2 susceptible lines that we used as controls. And then there were 13 genotypes that we used from this Wallier study that had implicated aflatoxin resistance. And some of these genotypes are what are called black skin varieties. They're not really black skin, but they have more of a purple seed coat. And you can see those in this picture. And then we used 2 other black skin varieties that were not in the Wallier study, but had some previous evidence of resistance. And these 2 varieties were Carolina Black 10 and Carolina Black 8. And then we used 2 chromosome segment substitution lines, these CSSL lines, which had wild peanut introgressions. And so the way that we designed this experiment is we sterilized and inoculated these seeds while controlling for seed health, temperature, and humidity in our incubation chambers. And we actually inoculated with a transgenic isolate of Aspergillus flavus, which produces a green fluorescent protein, so that we could visualize fungal growth on our seed coat. So all of this bright green light in the picture here is fungal growth on our seed coat. And then we ground up those seeds to extract aflatoxin, and we quantified the amount of aflatoxin using immunoaffinity columns and a Vicam fluorometer to get all this really nice aflatoxin data that we could then analyze through statistical analysis. And so just a little bit more about our inoculation procedures. We sterilized all the seeds under UV light for 30 minutes to make sure there were no contaminants. And then we inoculated with a standardized spore solution at 10,000 spores per milliliter. And we incubated those for 5 days in these really specialized humidity chambers, which were basically just airtight boxes, which had a little bit of water in the bottom of them. And then you can see all of the plates. This is post-inoculations. These are what the plates would have looked like. And so you can see some of that fungal growth on the seed coats. And then this would be what the seeds would have looked like under UV light. This is white light. So you can see all of the fungal growth very visibly here, but you can see it really starkly in the UV light photos because that green fluorescent protein is being produced. And so these were some of our results from our assays. So we split this kind of into 2 different sections. So on the left here are bar charts of the amount of fungal growth on our seed coats from all of the genotypes that we were looking at. And so you can see that's it was kind of similar for all of the genotypes. It's kind of all over the place, except for these 2 genotypes here, Carolina Black 10 and CS229, which is one of those recombinant inbred lines. They had very low levels of fungal growth across all of the experimental replicates. And then over here for this is box plots of all of the genotypes that we assayed for aflatoxin, and you can see that a lot of the genotypes that we tested were actually showing some low aflatoxin contamination levels, including that Carolina Black 10, which had low fungal growth, and CS229, which also had low fungal growth. And then these 2 genotypes here were the 2 genotypes that we used as susceptible controls in our experiment. And so you can see that they definitely were susceptible to contamination in this experiment as well. And so from some of the conclusions we drew from this is we had contamination resistance in 14 of the genotypes out of the 23 that we screened. That included the 4 recombinant inbred lines from the previous study, as well as both of the chromosome segment substitution lines that we tested, as well as that black skin genotype, Carolina Black. And then Carolina Black 10, as well as CS229, also had possible Aspergillus flavus resistance because they were demonstrating a lot of resistance to fungal growth as well. And so these are all new potential sources of aflatoxin contamination resistance that we can examine in the future. So some of the future work I think is really exciting for this because we can develop populations from some of these genotypes, and we can do some additional genotyping for potential aflatoxin contamination resistance. And then we can do some genetic analyses on these lines as well, which is already kind of in the works for those 4 recombinant inbred lines. And then the importance of this is really that we can identify and develop markers in the future for aflatoxin contamination resistance. That way we can breed for aflatoxin contamination resistance in the future. And so with that, I just wanted to acknowledge all of my lab members, my committee members, as well as the USDA, the Peanut Foundation, and Morris Wrigley for funding this work. And I can take any questions.
Thank you very much for the presentation. I have one here I'll read to you. This comes from 2 questions. Actually, one first from Dr. Soraya Bertioli. Sounds scary. Could you comment on how likely is consumer in the United States to come across peanuts contaminated with aflatoxin? Should the consumer in this country be worried, or are there enough checks and balances to ensure food safety?
Yeah, so I think definitely here in the United States, I think we're safe. Just throughout the peanut supply chain, there are checks everywhere basically for aflatoxin. When the peanuts come out of the field, they get a USDA grade, and that tests initially for any visible fungal growth on seed coats. And then as they move along the peanut supply chain, they have a lot of visual checks for aflatoxin and chemical tests for aflatoxin. So Vicam is really the company that does a lot of the aflatoxin testing for any of the consumer products for peanut. And then I know the shelling industry actually has different actual lighting spectroscopy machines that they actually check for aflatoxin on. And in the United States, I think it's 15 ppb for consumer products right now is the limit for aflatoxin, but in Europe and other places, they have even more stringent regulations. But it's definitely more of a problem to the consumer in places where they don't have as strict regulations on aflatoxin.
Okay.
But I don't think consumers are worried.
All right, next question is, why did you incubate the inoculated A. flavus for only 5 days? Secondly, what's the name of transgenic A. flavus, which produces the transgenic color?
Okay, so the first part of the question, so we incubated for 5 days so we did a lot of tests to see what the best way to inoculate was. And actually the first time we inoculated we inoculated in just like regular tissue culture plates, and we weren't getting any growth really even after weeks of incubation. And so we moved them to these humidity chambers where we had these airtight boxes, we had water in the bottom, then we had plates which act as platforms for the actual incubated seed plates so that they wouldn't get wet, but they would have a lot of humidity available. And we were actually getting a lot of aspergillus growth, like it was getting to the point where it was the Vicam machine for all of the aflatoxin that we were getting produced. We'd have to keep retesting them, diluting the solution and retesting them because we were getting too much aflatoxin being produced. And so we reduced the time from 7 days to 5 days, and we were getting a good measurable amount of aflatoxin on the seed coats.